Hi all. Now we are moving into another approach of microbial taxonomy or taxonomy in general. It is called a numerical taxonomy. This is a different approach, or it's a modern approach in which we use the computers and the programming and softwares for the analysis and identification and classification of organisms. And for this one, for the numerical taxonomy, we greatly depend on computers, and also we depend on certain area called bioinformatics, right? So the characters or the sequences identified will be, or the different data obtained will be provided to a computer, and the computer will do the rest of the analysis, and the computer or the softwares will give you the classification and identification of organisms, right? So based on that, it is easy to do the identification process or the classification process, right? So it's actually an operationally defined and numerically oriented procedure for taxonomic analysis, right? And this was introduced by H.S. Sneath and Robert Sokal, and they defined them as the grouping by numerical methods of taxonomic units into taxa on the basis of their character states, right? So here we are using the similar characteristic which we studied in a phonetic or phylogenetic system, but the difference is that we'll convert these characters into a machine readable format. That means into a numerical format, usually into 0 and 1. If a character is present, then it is given the value 1, and if it is absent, the value 0 is given. So each character is represented or is converted into the numerical formats, and these data is provided to a computer, and the computer will analyze, right? And accordingly you will get a value, or you will get certain coefficients or something, some value will be provided, and based on that we can say that this organism is having this much similarity with the other organism. So in that way, this numerical taxonomy is different from the rest of the system, that means phonetic and phylogenetic systems, right? And because of the great influence of computers, this taxonomy is otherwise also called computer assistant taxonomy or Adansonian classification. Okay. So different characters, like given like morphological, physical, chemical, metabolic, all the different characters also will be studied, right? And most importantly, the molecular studies are also being performed for numerical taxonomy. So there should be characters of around 100 characters should be analyzed for a reliable and accurate classification. Usually, the process begins with the determination of presence or absence of certain selected characters in a group of organisms under study, right? Or it may not be always a group. If you have a particular single organism, then you study, you observe for the characters, you study the characters, and you find that there are certain characters present in organism, and there are certain characters which are absent in that organism. So accordingly, you will compare this organism with an already existing organism. So you have two organisms, organism A and organism B. The organism A is the known organism, which is already studied and classified organism, and organism B is the unknown organism, right? And if you take some around 50 different characters, and we will study all the 50 different characters in the unknown organism, that means in the organism B, and already the 50 characters and their results are there with organism A. Okay, so what actually we do is we'll compare all the 50 characteristics, and we will say that the character 1 is present in organism B and is absent in organism A, and the character 2 is absent in organism B and present in organism A, and so on. So in this way we will compare, right? And altogether finally we will have a particular number of characters which are present in both organisms A and B. Right, there will be certain characters which are present in both the organism A and B, and there will be some other characters which are present in organism A and not in organism B, right? And vice versa also. That means there are certain characters which are present in B but absent in A, and there are also, there could be some characters which are absent in both organisms, right? So based on these values, we construct certain coefficients, right? Or the computer actually constructs certain coefficients. There are two types of coefficients widely used. One is the symbol matching coefficient, and the second one is the Jaccard coefficient, right? And symbol matching coefficient, or SSM, is denoted like a plus d divided by a plus b plus c plus d. And here a and d are, or the a b c d are, a is the character which is present in both organism, and b is the character which is present in a and absent in b, and c is the character or the number of character which is present in b and absent in a, and d is the number of character which are absent in both organisms, right? So by that we will get a value, and that is the symbol matching coefficient. And in the other one, the Jaccard coefficient, which is the widely used coefficient and which is little more reliable, and in which those characters which are absent in both organisms are not considered. That means d is not considered for the calculation. So the equation is simply converted into a divided by a plus b plus c, right? So this is the way how we calculate the coefficient. So once this association coefficient is calculated, this value actually represents, or this association coefficient is a measure of agreement between the characters possessed by these two organisms, or how much these two organisms are related will be denoted by this value. All right. Then both coefficients usually increase in a linear fashion from 0 to 1. If you have 100% similarity between organisms, the value will be 1. Right. Accordingly, if it is the value is 0.75, that means there are two organisms are 75% similar. All right. And once you get the coefficients, right, now what you have to remember the steps. You have an unknown organism, you studied this organism, you got the data, and the data is used for calculation of association coefficient. Then this association coefficient, once you obtained, is given into a similarity matrix or provided in a table, right? This data is arranged into a table, and this table is called similarity matrix, right? And actually in similarity matrix, the rows and columns represents organism. There are organisms like what many number of organisms are there, all of them could be compared each other, right? There will be a comparative analysis between each and every organism. So the rows and columns represents organisms, right? And we will provide the values, that means association coefficient value, to the concerned columns. Clear? So each value of this matrix is an association coefficient which measures the similarity of two different organisms. And in short, each organism will be compared with each and every other organism, right? So this will give you an idea. So you can see the bacteria 1, 2, 3, 4, 5, 6 is given in one axis, and the same organisms are given on the other axis also. And the values given are representing the association coefficient. So you can say the association coefficient between the organism 3 and organism 5 is 0.45, right? You can locate that, you can spot that value. So the relationship or the association coefficient between organism 3 and 5 is 0.45, right? And between 2 and 4 is 0.31. So in that way you can categorize. And the value for 3 to 3 is 1, because these two are identical, there will be 100% similarity, right? And the computer also use certain color codes also, right, for comparing the similarity, that is also given there. So looking at the colors, you can say that whether they are identical or non-identical. And after that, you go for the similarity matrix, this data is further summarized in a tree-like diagram, right? A tree-like diagram is also constructed in numerical taxonomy, which is called a dendrogram. And in the dendrogram, usually there will be an x-axis which is graduated in units of similarity. That means on one axis the similarity is provided, on the other axis the organisms are given, right? And the organisms in the two branches share so many characteristics that the two groups are seen to be separate only after examination of association coefficient greater than the magnitude of the branch point value. Right, that means we'll study the coefficients, we'll look at the coefficients, and the value of coefficients will give you the position of the branches. Right. When we look at the diagram, you will get a much clear idea. Right. So here you can see the organism 1 and 2 are almost 90% similar. Right, they are having a 90% similarity. And in case of organism 3, 3 is related to organism 1 and 2 and having a percentage of 80 similarity. So in that way these organisms are related. All right. So this is called a dendrogram, which is actually depicting the relationships of organism based on the value provided in the similarity matrix, and that value is obtained by calculating the association coefficients, and association coefficients are calculated based on the similarity between organisms in terms of their characters. So organisms with the greater similarity are grouped together and separated from dissimilar organisms, and such groups of organisms are called the phenons, or sometimes it's also called phenoms, right? And usually a phenon or a phenom will be given a value, a digit before that, like 70 phenon, 80 phenon. That means which represents the percentage of similarity. A 70 phenon is a phenon with 70% or greater similarity among its constituents. And usually when two organisms or a cluster of organisms with 80% or more similarity are often equivalent to a species. Right, when two organisms are having 80% or more similarity, then it is called a species. So that's about the numerical taxonomy. That's a brief discussion on numerical taxonomy, and hope you can understand that. So in short, it is nothing but this is a computer assisted taxonomy in which the different characters are analyzed, and all the characters are converted into the numerical formats. Then the computer will analyze, then there will be calculation of association coefficient, then these association coefficients are converted into similarity matrix, then the data from the similarity matrix are plotted into a tree-like diagram called a dendrogram. So that is the flowchart for this numerical taxonomy. Now the final part of taxonomy called identification. This is actually the practical side of taxonomy. This is the process of determining that a particular isolate belongs to a recognized taxon. And here actually this is the laboratory side. Here we study the different characteristics. Right. Various classical and molecular characteristics are broadly studied, and based on that we'll identify this organism. So in the next part of presentation we will see different characteristics or different criteria used for the identification process. So by that we wind up this part of presentation. Thank you for listening.